All right, thank you for that introduction. So the hypothesis we're addressing in this experiment is that pulse focus ultrasound induces calcium-dependent molecular changes in multiple mammalian tissue types. So to start off with some background, our lab has found in the past that targeting pulse focus ultrasound to the kidney or skeletal muscle results in increased mesenchymal stem cell homing to these tissues, resulting in downstream therapeutic response. And we've further found that this is a COX-2-dependent response, resulting in an upregulation of chemoattractants, including chemokines, cytokines, and trophic factors, and cell adhesion molecules. Through further in vivo studies, we have found that when you block the activity of voltage-gated calcium channels through verapamil, or knock down the activity of TRPC1, we are not seeing the same increase in COX-2 expression or the downstream mesenchymal stem cell homing, suggesting that both TRPC1 and voltage-gated calcium channels are necessary for this response. Additionally, since TRPC1 is a mechanically activated sodium and calcium channel that can also be activated through second messengers such as calcium, and voltage-gated calcium channel, as the name implies, is voltage-gated and conducts calcium, it was clear that calcium is very important in this response, and the downstream cascade is calcium dependent. We wanted to further investigate how the TRPC1 channels and voltage-gated calcium channels were interacting in order to create this calcium current, specifically what order they were being activated in. To do that, we looked in vitro model using fluorescent imaging at these ultrasound parameters. First, we confirmed that we see the same calcium-dependent increase in COX-2 expression in vitro, looking at TCMK1 kidney cells or C2C12 muscle cells. And you can inhibit this increase in COX-2 expression, as you can see in the FUS column, by loading the cells with BAPTA, an intracellular calcium chelator. We are then able to further look at further determine this mechanism using in vitro live calcium or sodium imaging, and we were able to look at relative calcium and sodium influx in response to various drug and genetic manipulations. So what we end up finding is that calcium influx is dependent on voltage-gated calcium channel activation. By blocking voltage-gated calcium channel by blocking the voltage-gated calcium channels using verapamil, we see a cessation in calcium influx, similar to what we see using an intracellular calcium chelator, BAPTA. However, we also find that this calcium influx is dependent on sodium influx. And we did this by reducing the driving potential of sodium by doing a partial cesium replacement in the extracellular liquid. We further find that this is dependent on TRPC1 through a partial TRPC1 knockout. Finally, we find that this upstream sodium influx is occurring through TRPC1, as a partial TRPC1 knockout does significantly decrease the sodium influx. So the takeaway from this slide is that while voltage-gated calcium channels are necessary for the downstream calcium-dependent response, it is secondary activation to the TRPC1 and then the local depolarizations through the sodium flowing through TRPC1. Whoops. As mechanically sensitive moieties like TRPC1 are very common throughout multiple tissue types, we wanted to see whether this was a sustained response. We see an analogous calcium influx in mesenchymal stem cells, B16 melanoma, HCN2 neurons, and 421 breast cancer lines. We also see a similar calcium-dependent COX-2 increase in expression as we had seen earlier in kidney muscle, which is inhibited using the intracellular calcium chelator BAPTA. To further demonstrate this is a robust response, we see this in cell lines that are derived both from the mesoderm and the ectoderm, as well as originating from both mouse and human lines. So in conclusion, we see that there is a similar calcium transient initiated across a wide variety of cell types, although the kinetics and magnitude of response vary. COX-2 is consistently upregulated in response to this intracellular calcium signaling. And in the future, we want to determine whether TRPC1 or other mechanically sensitive channels are activated in these various cell types. And we also want to further investigate how downstream calcium processes may be amplifying this cytosolic calcium influx, such as through calcium-induced calcium release or store-operated calcium entry. Thank you for your attention. I'll take any questions.
Questions?
In pulse-focused ultrasound, there are so many parameters to choose. It's an amplitude, a frequency, pulse length, duty cycle. Like, what was the motivation to choose these exact parameters, and which one is the most important, if it's known?
Yeah, thank you. So we chose these parameters based on the fact that we've used 4 megapascal and 1 megahertz in the in vivo work that I covered at the beginning slide. It's very interesting that you mentioned something like duty cycle. We're not entirely sure whether we would see a transient response similar to what we see, what I've observed, or if we would see a sustained influx, and that's something we're looking forward to in the future.
To follow up that question, I would suggest to look at mechanical deformation of the cells or tissue as a better indicator. Your acoustic parameter could vary significantly. It's really the mechanical response of the target cell tissue to the ultrasound that triggers the calcium response. So have you measured deformation of cells through experiment?
We haven't done it in vitro, but I believe we have done it in vivo. We've looked at the acoustic radiation forces, yes. That's actually a suggestion that I got for future studies, is to use a different channel to look at possible decompression of the cells as well.
I guess one more follow-up to this question is, would you expect to see any intracellular or extracellular cavitation in those cultures?
We do see endogenous cavitation at 4 megapascals in vivo. It's not something we've measured in the tissue dish, but I wouldn't eliminate it as a possibility.
Great. If no more questions, thank you.